Greetings of the day. Hope you're all doing well. I am Miss Retty Shaji, Assistant Professor, Forensic Department, Kristu Jayanti College, Autonomous Bangalore. So the subject that I'm gonna take today is forensic serology. As you all know, from a crime scene, it's easy to get body fluids or tissues. The most of the samples that we encounter from a crime scene is body fluids. So, to identify those body fluids and relate it to a crime scene with the suspect is what serology department does. So, today the topic that we are going to handle is ABO blood grouping. So, the most of the sample will be blood droplets from the crime scene. We know that blood is a fluid connective tissue which is very essential for our body in transporting nutrients as well as the oxygen and removing the waste materials from our body. And our blood mainly composed of two main components. One is plasma, the other one is the blood cells. So 55% of our blood is composed of plasma. And the rest 45% is composed of the blood cells. In that we have red blood cells, which is the erythrocytes. Then we have WBC, leukocytes, and platelets, thrombocytes. The most of the, out of 45, 44% is made of the RBC, and rest WBC and platelets is less than 1%. So we'll begin with the history of ABO blood grouping. So the father of blood transfusion, or the person who has discovered the ABO blood grouping, is an Austrian scientist named Karl Landsteiner. So it started in the year 90s, early 90s. The Karl Landsteiner has led to the discovery of ABO blood grouping. Earlier times, in ancient times, all the people believed that everyone is having the same blood type. So when they started doing blood transfusions, they found that some of the blood transfusions were successful, where some other were failure, which has led to even fatal conditions. So Karl Landsteiner started his experiments on studying the blood transfusions, and he found that certain molecules which are present in our plasma is agglutinating or forming clumps with certain molecules which are present on the surface of our RBC. And so he found that the molecules which are present on the surface of the RBC is known as the antigens, or the proteins or the molecules present on the surface of the plasma is known as antibodies. So, based on what type of antigen is present on the surface of the red blood cells, he has categorized it as 3, A, B, and C. Later, C was renamed as O, which stands for on. In German, it means without. So, any person with the blood group A means he is having A antigen on their surface of the red blood cells. And a person with B blood group is having the antigen B on the surface of their red blood cells. And a person with blood group O is not having any antigens, that is, the absence of both A and B antigen on the surface of their red blood cells. And in 1901, the Karl Landsteiner has published a paper, and he named the paper as Agglutination Phenomena of Normal Human Blood. In the paper, he has described in detail about the blood groups, the antigens, the antibodies present in our blood. And later in 1902, he discovered one more blood group, that is AB blood group. That is, any person with the blood group AB is having both A and B antigen on the surface of their red blood cells. Later, two of his other colleagues, they are Alfred de Castello and Adriano Sterli, they have combined all the principles that is brought up by the Karl Landsteiner, and they have characterized ABO blood grouping system, and they also explained about the inheritance pattern. So the inheritance pattern of the ABO blood grouping system follows the Mendelian pattern. And later, from 1910 to 1920s until now, in all the healthcare professionals and in all the hospitals, before they do the transfusion, they will also analyze the blood group. So this is the history of our ABO blood group. Next, look into our biosynthesis of ABO antigens. So, we know that in humans we have 23 pairs of chromosomes. So, in chromosome number 19, we have a gene called FUT gene, which stands for fucosyltransferase. So, mainly chromosome number 19 has two FUT genes, FUT1 and FUT2 genes. So now we are talking about the antigens present in our red blood cells. So we are focusing on FUT1 gene, which is responsible for it. So what is the role of fucosyltransferase? Fucosyltransferase helps to transfer a polysaccharide, which is the fucose molecule, to the glycolipid end of the erythrocyte. So we are having our erythrocyte, which is the RBC. And consider there is a glycolipid in our RBC, and the fucosyltransferase is going to attach the fucose molecule to the end of the glycolipid, which is present in our RBC. So once the fucose molecule is attached to the end of the erythrocyte, it's going to get synthesized O or H antigen in our RBC. So every individual is having O antigen in their body. So how it is getting synthesized in our body? When in the chromosome number 19, the gene, the FUT gene, is going to transfer a fucose sugar molecule with the help of a fucosyltransferase enzyme, which will synthesize the O antigen in our body. So now, from the O antigen, how we are going to have A and B antigen in our body? So we know that we are going to inherit certain alleles from our parents. So some people will inherit A allele from their parent, some gonna inherit B alleles, some gonna inherit both A and B alleles. So based on which allele you're gonna inherit from your parent, based on that you're gonna get A antigen or B antigen in your RBC. So now let's look into that. So we are having O antigen in your body. So if consider that you are having A allele in your body, which is inherited from your parent, that is one from your mother, one from your father, you are having A allele in your body. If you are having A allele in your body, it's gonna produce an enzyme called A transferase enzyme. If A transferase enzyme is present in your body, it's gonna transfer an enzyme called N-acetyl galactosamine transferase will be produced. So it's gonna transfer N-acetyl galactosamine to the O antigen. Okay, so a molecule or a sugar molecule called N-acetyl galactosamine will be attached to the end of the O antigen. If N-acetyl galactosamine is attached to the end of the O antigen, you're gonna get synthesized A antigen in your RBC cells. If in case you have inherited B allele from your parent, B allele gonna produce B transferase enzyme. That B transferase enzyme will help in producing galactose transferase enzyme. So what is the role of galactose transferase enzyme? That gonna attach or transfer a sugar molecule called galactose to the end of the O antigen. If galactose is getting attached to the end of the O antigen, then you're gonna get B antigen synthesized in the surface of the red blood cells. Okay, so A antigen and B antigen differs based on what sugar molecule is attached to the end of the O antigen. If N-acetyl galactosamine is attached to the end of the O antigen, you're going to have A antigen in your RBC. If galactose sugar molecule is attached to the end of the O antigen, you're going to have B antigen in your body. If both transferase is present, that is you have inherited both A allele and B allele from your parent, and it's gonna produce both A and B transferase, which encodes for N-acetyl galactosamine transferase as well as galactose transferase. So they're gonna do both the work, that is it's gonna attach N-acetyl galactosamine to the end of O antigen as well as galactose molecule also to the end of the O antigen. So you're gonna have both A antigen and B antigen in your RBC. So you're going to have the blood group AB blood group. Consider you have inherited O allele from your parent. If O allele is inherited from the parent, it's going to undergo certain mutation, terminal mutation, and there is no change gonna happen to your O antigen. So there will be, it's gonna lack both A and B antigen, but you're gonna have O antigen in your body, and the blood group that you're gonna have is O blood group. So if you're having A antigen in your RBC, you're gonna have A blood group. B antigen in your RBC, you're gonna have B blood group. Both A and B antigen in your RBC, you're gonna have AB blood group, and O antigen, that is gonna have O blood group. Now, let's look into the genetics of ABO blood group. So, from the characterization done by the Karl Landsteiner, we know that there are 3 alleles present in your body. One is A, B, and O alleles. Okay. So, based on those alleles present in your body, there are certain genotypes that you're going to have and certain phenotypes you're going to have. So, if you're having both A allele, that is a homozygous A condition. Homozygous means you're going to have 2 identical alleles in your body. That is, you're going to have both A alleles in your body, that is homozygous A, which is going to represent A blood group, that's your phenotype. If you're having both B allele, that is homozygous B allele, which is the genotype, you're going to inherit the phenotype as B blood group, B phenotype. In the same way, there is a heterozygous condition, that is, you're going to have 2 different alleles. So, for A blood group, A phenotype, you can have A allele as well as O allele. That is a heterozygous condition. Okay, so for a blood group A, you're going to have 2 genotype, homozygous A condition and heterozygous A condition. For homozygous, it's both AA, and in a heterozygous, it is A and O. Same as in B, you're going to have 2 genotype condition, one homozygous and one heterozygous. Homozygous condition will be both B allele, and for a heterozygous, it will be one B allele and another one be O allele. Okay, in AB blood group, it's a codominance allele. That is, both A and B allele will be equally expressed. Okay, in O blood group, it's only one genotype, that is both homozygous O alleles. So this is about the genetics of ABO blood group. Now, secretors and non-secretors. So some individual is going to be secretor, some going to be non-secretor. So we know that our blood antigen is present on the surface of the RBC. But in certain individuals, these blood antigens, that is A, B and O antigens, will also be present in other body fluids like saliva, semen, etc. Okay, so those individuals in which the blood antigen is also found in, other than the RBC cells, they are also found in the other body tissues or secretions, they will be known as secretors. And certain individuals in which the blood antigen is only found on the surface of the RBC cells, they are known as non-secretors. Now, what will determine a person to be a secretor and a non-secretor? It is also based on our FUT gene, which is present in the chromosome number 19. So, I have already said you have 2 FUT gene in the chromosome number 19. One is FUT1 and FUT2. So, when we were talking about the antigens present on the red blood cells, we were focusing on the FUT1 gene. So, FUT1 gene helps in producing the antigens in your red blood cells. Mainly, they are active in the tissues of mesodermal origin, that is in your muscles, in your bones, in the red blood cells. This FUT1 gene is more active and they are helpful in producing the blood antigens. Whereas FUT2 gene, they are active in the tissues of endodermal origin, that is in your secretions, that in your salivas, in your gut, in your internal organs. These FUT2 gene is active. That is, FUT2 gene is responsible for producing the blood antigens in the tissues of endodermal origin. Now, let's look more into the secretors and non-secretors. So, FUT2 gene should be responsible or should be active and functional in a person who is a secretor. That is, they are responsible for producing the blood antigens in your secretions. So, once FUT2 gene is active in your secretions, fucosyl transferase enzyme will be producing, and we know that the role of fucosyl transferase is that they are going to attach the fucose sugar molecule to the O antigen. If O antigen is being produced and if you're having A allele and B allele inherited from the parent, based on that you're going to have A and B antigen in your body fluid also. Now in a non-secretor, they are also having FUT2 gene, but it is non-functional. That is, they are undergoing mutation at the amino acid position 143. So since they are undergoing mutation, it's going to produce a truncated protein, that is a short fragmented non-functional protein. So since you are having a non-functional FUT2 gene, what happens? There won't be any O antigen synthesized and no fucosal transferase enzyme will be there. So no A and B antigen in your body fluid. Okay, so that is the difference between secretor and non-secretor. So FUT2 gene is responsible for a person to produce the blood antigen in their secretion. So now just an exercise for you. So antigen A was found in the saliva of an individual. So he will be a secretor. And if A and B antigen is found in the plasma, he will be a secretor or non-secretor. He will be a secretor because other than the red blood cells, they are present in the secretion, which is the plasma. Now, third one. A and B antigen was found in the red blood cell but not found in the plasma or semen. So he will be a non-secretor because it's only found in the red blood cells. So a just summary of what we have just learned through now. So we know that ABO blood group is discovered by Karl Landsteiner. He is known as the father of transfusion medicine. And it is the blood antigens or the blood groups are synthesized or categorized by glycosyl transferase enzymes. That is going to add a sugar molecules, either N-acetyl galactosamine, which is given by the A allele, to the H antigen or O antigen, and galactose sugar molecule, which is given by the B allele, to the H antigen, which is going to have B antigen in your body, and O allele, it's going to undergo a mutation, so a non-functional enzyme will be produced, so there is no change going to happen, so it remains the same. And the difference between secretor and non-secretor, the secretor is going to express the blood antigens in their body fluids and the secretions. A non-secretor won't be having ABO blood antigens in their body fluids. It will only be present in the surface of the red blood cells. And it is mainly controlled by the gene FUT2 gene, which is encoded by the chromosome number 19. So I hope you all had a great section. Thank you.